Well, I want to thank everybody for joining us this morning. So on behalf of Metabolon, I'd like to welcome you to a deeper understanding of COVID-19 phenotypes through metabolomics, a panel discussion. My name is Patricia Sheridan. I am a senior study director in the biological development group at Metabolon, and I will be the host and moderator for today's event. Before I introduce our presenters, I'd like to provide some brief instructions. The presentation's been muted, but we welcome as many questions as you have that we would like to answer during the Q&A session at the end of the presentations. To submit a question, please open the Q&A window at the bottom of your screen, and you can submit that question anytime during the presentations. Now I'd like to introduce our presenters for today. They are Dr. James Heath, Ph.D., President and Professor at the Institute for Systems Biology, and Ujjwal Neogi, Associate Professor of Virology in the Department of Laboratory Medicine at Karolinska Institutet and an Adjunct Associate Professor at the University of Missouri School of Medicine. So I'd like to welcome everybody. Thank you so much, both of you, for taking time. I realize it's the afternoon in Sweden and very early in the morning in Seattle, but we really appreciate you joining us. And so no further ado, Dr. Heath, if you would like to start, that would be great.
Sure. Thank you so much, Pat. Let me share my screen. So I'm just going to say a few words about a study that we've just recently published this in Cell. I think it comes out in the print version in about 2 weeks or something like that. And it's an ongoing study. So we published the first part of it, and I wanted to particularly we had some very interesting findings with relevance to metabolites in COVID patients. And so I wanted to just highlight that as a part of this webinar. So this is it's a 200 patient study. And what we did in this study was we looked at serial draws from about 140 of those patients, really spanning the range of disease severities. So from asymptomatic or mildly symptomatic all the way up to severely ill. And then we did almost everything you can think of on these patients. So a really comprehensive analysis. And this gives you sort of a feeling of the clinical picture of the patients we studied. And so these are what are called swimmer's lanes plots, where the color that you see here, as it goes from light colored to darker red, that indicates the severity of the disease according to the WHO ordinal scale of severity. So the redder you are, you're likely intubated, and if they're the most red, you're on organ support as well. But if you're green, you're likely convalescent and possibly even at home. And then lots and lots of clinical measurements that we do on these patients, and we didn't really find too much new in these clinical measurements. But what we did find that I think was fundamentally new, and I think has some implications for therapy, is sort of summarized in this single plot here. So this rather complicated looking plot is what's called an interaction plot. And so what I've shown, we measured on the order of the whole metabolome assay of 1,000 or so metabolites, about 500 plasma proteins, and then this is some clinical measures. So the proteins are here in the orange, a few clinical measurements in the red, and then metabolites grouped as classes of metabolites. So for example, this lysoplasmalogen might represent 10 or so different lipids, but they're represented as classes. And when I have these connections between them, if it's a dashed connection, that's an anti-correlation. If it's a solid line, it's a correlation. But what I've limited this to is only those correlations that are strongly affected by disease severity. Okay, so that's the disease severity is the interaction. And I show a representative interaction here, which is one of these lysoplasmalogens with this pro-inflammatory protein IL-6. You see that IL-6 has many, such connections coming out. So this lipid in healthy or mildly infected patients has a large range of values. It's a typical lipid that has certain levels in the circulated in the blood. This is a lipid that's important for cell membrane construction. In fact, most of these lipids you see pop up are. And as disease severity increases, you have inflammation increasing, which is this axis here, but this lipid level profoundly drops and stays dropped. And this type of behavior, increasing inflammation with loss of lipids, loss of amino acids, it turns out loss of xenobiotic metabolism, which is basically the metabolism of food products, is profound. And just to show I didn't cherry pick this plot here, here's just a whole bunch of plots. And so what, and this is like a lot of more lipids associated with cell membranes, and then xenobiotics. And here the axes are changed. So the lipids are on the x-axis and the proteins are on the y-axis. But it's a phenomenon that happens that suggests that as disease severity increases, there is a stronger and stronger inflammatory response, suggesting that you're demanding a stronger anti-immune response against, or antiviral response from the immune system. At the same time, you've depleted the blood of nutrients from which to build that response. And so it's a frustrated environment. And then we did a lot of single cell analysis. I'm not going to talk about that, but what we found is that and some of this has been found before, but we found that for virtually every single immune cell class, at the stage where you switch from mild to moderate disease, so at the stage where this turns the corner, you see some novel or dysfunctional immune cell class emerge. And it could be a cytotoxic CD4 cell. It could be cells that have proliferative and exhaustive characteristics, et cetera. And so the picture that emerges out of this is that you've got this sort of stressed plasma environment of pro-inflammatory, depleted nutrients, out of which you've got these different dysfunctional immune cells. And we looked at all the patient data, we actually correlated it all together to get one overriding gene module, and it basically all of these things emerge from that one gene module. And what it suggests, it suggests a couple of things. One, we don't really know if this is unique to COVID. I think, as we've been studying COVID in such great depth over the past several months, we're finding that we are learning a lot about COVID as an infectious disease, but we don't know. We're finding what we don't know about things as simple as influenza or other infectious diseases. But it also suggests that there's possibly non-pharmacological interventions, such as supplements and things like that may actually be very useful for patients. It also suggests that moderate disease, before patients are intubated, it looks almost identical to severe disease. Severe disease is just moderate disease with the volume turned up. Moderate disease is very different from mild. And so moderate disease is the best place to apply therapeutic interventions because you have a better chance of response, and it effectively has the same symptoms, the same immune cell responses, et cetera, as some more severe disease where patients are harder to treat. And with that, I'll pass the wand to Ujjwal.
Okay, thank you, James. It's a nice introduction, actually. So we also do more omics study or integrative omics, but we go far beyond because my group strength is to the functional, because the nice question that Jim has said, this is because of only COVID, maybe influenza or other thing. So when we started our study, our main, that time in Sweden, we not tested the mild, so only it's hospitalized patient. So we categorized the patient as a hospitalized mild based on the oxygen consumption at the time of sample collection. So we uh, we categorize hospitalized mild and hospitalized severe based on 4 liter per minute oxygen consumption. So, and then we did metabolomics and transcriptomics over the time, and we also have an HIV COVID negative samples. But we started with the metabolomics first because we want to know more about the what kind of metabolic changes happen when there's a COVID infection, SARS-CoV-2 infection. So we did metabolomics, proteomics, and transcriptomics, and then we further gone to the validation, like using qPCR or Western blot or FACS. Apart from that, we also started looking into infecting the cell models. Like we have infected different cell. With our first paper is on HU7, but we also infected Kalu3, Kako2, like of different origin, and also my collaborator at Public Health Sweden, they have infected organoids and then do some proteomic study to identify the mechanism. So if you go to the next slide. So what we have initially did in the HU7 cell, so we did a proteomics, so we infected with different concentration of the virus or different MOI of the virus and different time point to mimic that, okay, low to high viral load, and then how the host response happen. And then we use transcriptomics and proteomics over the time, and then use co-expression network analysis and other analysis to find out. So what we have observed that one of the striking feature is the mTOR signaling pathway or HIF1 signaling pathway. So it seems that virus is using the HIF1, HIF stabilization, and they are using that one for its own gain. And as we know that HIF1 is one of the modulator of the glycolysis pathway or the central carbon metabolism, because HIF1 signaling the HIF1, when it not degraded, it translocate to the nucleus and then it activate HIF targeted genes. And most of the genes are for that is linked with the glycolysis. Then we did a patient metabolomic study with the 4 group that I mentioned, and we are mainly interested in central carbon metabolism. So I just showed some figure of lactate. So interestingly, this is high in COVID mild patient, but little bit lower in COVID severe patient. It could be because they already has a dysfunctional immune system. Pyruvate citrate is low. That, as Jim also showed, that lots of things are going down, but glucose is high, mannose is high, and what I believe James paper also shown. So now what we observe when we target it, so we are mainly interested in host targeted therapy for infectious disease. So we used a drug called MK2206, that is AKT inhibitor. And then we saw that there's a low viral load compared to the non-treated. So what we have hypothesized the distribution of PIK3 AKT signaling, which could be HIF mediated, but we still don't know though the virus is using the HIF. And there's a nice study came from Brazil. They showed that SARS-CoV-2 initially stabilized the HIF for translocation of the nucleus, but our data was not like that. And then how that is linked with center carbon metabolism. And finally, can the central carbon metabolism biomarker can act as a modulator, and or we can give a non-clinical, not drug intervention, non-COVID intervention, or say nutrition or some metabolic thing for the patient, and then cope up with the disease severity until we have a proper drug as well as the vaccine. So my research group also work with, actually we started this work with HIV and followed by Crimea Congo hemorrhagic fever. But it's importantly what happened like with the SARS-CoV-2. It seems that all these viruses, RNA viruses, has used this central carbon metabolism for its own gain. So we would like to understand more, because we also now clearly seen that this utilization of central carbon metabolism is tissue specific, it only happens in the lung cell, but not in the intestinal or other cells. So we hopefully will submit the paper soon, maybe next week, and we'll take from there for the next stage. Like our main interest is host targeted therapy. That is how we can boost up the immune system of the host that can compete with that can take care of the virus by themselves. Yeah, so I'm with that. I'm done.
Wonderful. Both of those are so much interesting data. So I wanted to start, Ujjwal, with you just off that last comment. You said you were seeing changes in the central carbon metabolism in the lung cells, which are one of the primary sites of infection for the virus, but not in the intestine. And it is believed that SARS, or the COVID-19 virus, the SARS-CoV-2, does infect the intestinal cells. Do you have an idea about why there might be those differences in the response to in the different cell types?
Yeah, so we know little bit about that, because not only intestinal, we also used other cell line also CACO2, because intestinal is the organoid, intestinal organoid, and cell line we use CACO2 and HU7. We obviously the response is different because it's a lung disease. So what our working hypothesis is, like it's the metabolic reprogramming mainly happening in the lung primarily, then it's coming to the circulation, and then if the severe cases is going to the other organs, and then having a different, and also the other organ, though some studies has shown that they are able to infect in vitro the blood cells, we are not able to infect that. So even the the infection profile is maybe much lower compared, maybe in very severe cases. And in our patient data, we don't have any ventilated patient, because metabolomics has a big impact about the present condition of the nutrition. So we try to pick up the samples, like there is no additional drip or something like that, and no other medications at least 12 hours before, and there is something like that, so that we can have a proper, so we want to mainly understand is COVID specific or not.
Actually, that leads nicely into a question from the audience for Jim. To what degree can you correlate the depletion of the nutritional metabolites to the diet of the sick patients? And what that might mean for interventions?
Yeah, that's a great question. And it's, in fact, it's exactly what we're trying to understand right now. So as you can imagine, many patients, at least the more severely infected patients, are going to be, they're intubated, they're going to be on a pretty minimal diet. You burn tons of calories when you are basically feverish. You can lie in a hospital bed and burn 3,500 calories in a day. So although we do see this onset at moderate disease, in which patients are going to be a little bit better off. I think what we're trying to understand is both what are possible dietary interventions, because I think there surely must be, as well as we're doing a bunch of measurements on flu, sepsis, Ebola, other viruses, infection disease right now, to try to get a grip on, is there anything about this that is unique to SARS-CoV-2 that can be addressed uniquely here? And if not, then one can begin looking in the literature. But I would be happy if there are dietary specialists out there that have a deep understanding of metabolism to reach out and talk to us. We've been trying to talk to a lot of folks, and it's a field in which sort of molecular details of metabolism and the sort of observations of dietary supplements, et cetera, have not really met, and at least for this pandemic, and I think it's an important area.
Ujjwal, back to you. What do you see as the next steps based on the studies that you have performed, and how might you continue to use metabolomics in these studies?
what we are trying to mainly understand, so I am also talking to those who are doing vaccine, because one important factor, I believe as a virologist, we are forgetting the virus itself, because virus use host metabolism for replication. And it's, for example, it's use glycolysis. Most of the viruses use glycolysis or glutaminolysis. Some use, and particularly for this envelope virus, they use N-glycan biosynthesis for its own gain. And we can clearly see both from Jim's study, our study, and a study from metabolomic study from China, that the mannose is high in the plasma. So that means it's secreting. And we treated the cells with mannose and then infect the virus. We didn't see any major changes in the viral replication. So we would like to know from where this mannose is coming. And second thing is, when mannose is high, there's a high possibility of the activation of the complement cascade, this mannose binding lectin pathway. So now we have seen the several patient who has blood coagulation thrombosis. So that could be highly linked with this mannose or mannose binding lectins and then activating blood. And even people are like they are released from the first few patients from Sweden actually released from the hospital, and they have complications, and then there's not much time left for them to save. Second thing is, we are also working to understand this happy hypoxia.
Mhm.
That is, like, we know that some people have very low oxygen saturation in the body, but still they are working. So that means it's also like some way in any lung diseases, hypoxia will be a outcome, and whether the virus is utilizing that stage to go for more rapid replication, or so you would like to understand. And the final thing, I was just talking to our collaborator who are working with the vaccine in FEDET. We also want to know, so they did a vaccine study and FEDET with their vaccine candidate. And then we would like to know what kind of changes are happening in the FEDET in terms of vaccine thing. Hopefully we will come up with. And as my focus, my lab focus is mainly on metabolomics towards the protranscriptomics. So we will start with the metabolomics. So that's, I think we will keep the focus of the lab for the COVID metabolomic studies.
Excellent. Um, Jim, the ISB study, if I am correct, is a prospective study so that people are being continuously sampled or called back.
Yeah. We're looking at it's ongoing. Yeah.
Do you have any insights right now into kind of what they're calling the long haul phenomenon? So people who have been infected with COVID and who months later still are experiencing either some kind of mild fog, shortness of breath, the symptoms that just seem to linger for a very long time.
Yeah, we're just beginning to resolve what at least appear to be some of those effects. So we've looked at patients a couple months out from the initial infection and tried to understand how different patients recover differently. And what we find is that there's something like 4 major pathways to recovery from the disease. And one of them appears to be what one might think about as these long haulers. And it's about, and I say that because 2 things. One, during the sort of acute form of the disease, as I mentioned, we see all of these various novel and dysfunctional immune cell phenotypes pop up, presumably coming out of this stressed plasma environment, but at least correlating with the stressed plasma environment. Several of those phenotypes persist in about 15% of the patients. And so there's a consistent immune dysfunction that seems to continue. And the percentage of patients that we see relative to what at least are been reported as long haulers is about right with that have those types of symptoms. So we haven't yet done a full analysis yet, but that's it. Basically, it's our full-time job right now is to try to resolve that. But hopefully in a month or two, we'll have a deeper insight. And we would like to know, does this plasma environment recover? And maybe it doesn't recover. Maybe that's why you see these immune cell phenotypes persist. We actually don't know that yet. But we'll also probably need to be looking out to 6 months to really resolve some of these phenomena as well. And there's other diseases for which long haulers have been reported. And in fact, it's been skeptically treated and sometimes by the scientific community. But one of those is Lyme. And we have a series of Lyme samples from patients that we are doing a side-by-side comparison with COVID, because I think there's actually a lot of similarities between what we see here. And I will also say one thing that probably relates to long haulers, but we don't know yet, is that in immunology, one has this picture or just an immunology textbook of how your immune system is supposed to respond to the virus. And your virus basically attacks tissues. Those tissues get eaten by your innate immune cells. Antigen gets presented. You get T cells and then B cells, and pretty soon you know about the virus and you're immune. That classical picture seems to happen in only a subset of the patients, maybe 30-40% of the patients. And the other patients are responding to the virus in very different ways. And so once again, and we've done a sort of a survey study that, and I don't want to say too much about it because it's still immature, that says that this is actually pan-viral. It's not just SARS-CoV-2, it's actually other viruses as well. But, all of this stuff is just highlighting just how much we don't know.
Yeah.
Even as we learn a lot.
Right. So Ujjwal, as you mentioned, I believe your primary focus of research prior to the COVID-19 outbreak is HIV. Can you address a little some of the similarities or differences you're seeing in the data between those types of studies?
Yes. So one thing for HIV, what we are working with mainly towards cure. And then a few years back, there's a nice study from Eric Verdin's group that mTOR mTOR pathway. If you try to intervene with the mTOR pathway blockers, and then there's a activation of the latent virus so that it can clear the cells and then we can achieve. But that strategy was called shock and kill, but this don't work that much. So when we did our first study, when we infected the cells with the SARS-CoV-2 virus, and we did, without knowing anything, we did transcriptomics and proteomics, and what we have observed, this is the primary pathways which was there in HIV has popped up. So we then immediately, because we have all the antibodies and everything standardized, we went for that and we show, but the mechanism could be different, but at least we know that SARS-CoV-2 also use like Heath signaling pathways, which basically linked with the metabolism. One thing what we have observed in long-term HIV patient, like those who are on 15, 16 years on treatment, that glutamate metabolism is one of the factor. High glutamate in the plasma. And we can see the similar thing in COVID patient also. So high glutamate level. So there is a shift between glycolysis, glutaminolysis during the acute COVID-19 compared and the chronic HIV patient, like those who are on therapy for a decade or more. So these are some similarities that we have observed. And we also did transporter expression analysis, and we have observed that there's some change in some cells. I am not mentioning the cell right now, where there is a change in the transporter expression. So that means it's going out of the cells very rapidly, so, and which is giving higher sugar content or carbohydrate concentration in the plasma. So these are the few common features between chronic treated HIV patient and the acute COVID-19 patient.
Right. Kind of a step back question. And both of you presented data that was multi-omic and clearly involves a lot of people. So outside of obviously the clinical staff that are needed to procure the samples, what type of researchers are involved in these studies that deal with these multi-omics? And it's the Institute for Systems Biology, says in its name, really gets down into the systems biology to answer these complex questions.
I'll take a stab at that first. So, as you can imagine, integrating all of this data and to give you at least some physical picture of disease and disease progression that relates to how you treat patients, it's a significant computational task. It's also, so that we have a lot of computer scientists that are working on this. And so on each of the patients that we're looking at, we're collecting their whole genome plus everything else practically. And it sometimes to analyze a single patient data set, you can have a pretty large computer chugging away for a day or so to spit out a result. And the other thing that I think is really important here is high quality measurement science. So one thing that we do with our metabolite and proteomic data is that we have 6,000 healthy individuals for which we have this same data, and we can compare as basically age, gender, ethnicity, et cetera, matched controls. And whenever we do a panel of metabolite or proteomic assays, we stick the same control plasma into each of those new samples to allow us to do direct comparison. And at the moment, we're bringing together a data set from Penn and from Sloan Kettering using these types of measurement analytics and common controls, which allow us to do now large-scale data integration. So measurement science is really important. And then the last thing is that having a lot of data is not the same as actually having much knowledge. And so there's a lot of work that just goes, in fact, I would say understanding the fundamental biology is very, very challenging here. And when you're doing transcript, and I'm sure Ujjwal will agree with this, when you're doing transcripts or proteins, you always have the genome that you can connect back to. It gives you some sort of a fundamental connection. When you're doing metabolites, you don't. It makes it it is actually really challenging to connect metabolites back to the basically the proteins and the genes that are processing those metabolites or that are dysregulated. You can only sort of infer those. There's not direct connections usually. So that's, I say, a piece of biology that is rapidly evolving, and this pandemic has forced us to more rapidly evolve it.
Ujjwal?
Yeah, so I will add, I echoed with Jim thing, but we also have a great clinician who are actually working in the this COVID world. So because this project, the patient selection, because we have a challenge because there are several studies which use ventilated patient, and initially in March, April, WHO category was not that developed because it's very arbitrary. High flow oxygen, low flow oxygen, but what is the measurement of high flow oxygen? So my study is actually primarily discussed with the clinicians. So we have a great infectious disease biology, and we also had a challenge that time because Sweden do not even test in the beginning those who are not reaching to hospital. So my study is actually a combination of clinicians. So they defined, okay, we will define mild or severe based on what we see in the clinic. And then based on, that's why we have categorized mild and severe based on the requirement of the oxygen, not any other arbitrary. And because we have to exclude for metabolomics, as Jim said, that there is no other way to check whether this is disease COVID oriented, or it's some lifestyle or other thing. And also there is lots of old people in the cohort, so that there is a change in their metabolite profiling. And then we have excellent nurses, because we try to minimize the pre-analytical errors, so that, and the definition, because what we have also done a study with Italy, where patient was categorized based on the type of marks their doctors are referring, like nasal cannula or vent mask or CPAP. But what we have observed at the end is very clinical decision, because there's not much difference between these things. So rather, as Jim said, there's a measurement science, which is important. So we measure our severity based on the requirement of oxygen. The second part is my group as also has immunologist, as well as cell and molecular biologists, because lots of, and virologists, because we have to work in biosafety level 3, and there are for the infected cell, because we just don't we started with the infection assay,
Right.
not to go directly to the patient. And then obviously computer science. So I have 4 computer sciences on these projects there. Now we worked with HIV, now we stopped, and we focus fully on COVID-19 for the last 6 months. So yeah, so it's a mixture of clinician, cell molecular biologists, immunologists, and computer science, including virologists.
So one, I would encourage more questions from the audience. But we have another question from those watching. How do you think, this is for both of you, vaccine development will impact COVID research? Do you think it will shift the focus away from other therapeutics? Or do you think things will, continue at the pace in the kind of same veins that they're moving right now? Jim, if you want to.
Okay. Well, I think it's certainly as soon as vaccines are broadly available, the urgency of some of this research is going to diminish, for sure. I think what's other than, I think that probably the long hauler issue is probably going to be with us for a while, and that's going to take a while to understand. I think there's a lot of ways you can look at what we've learned or are learning. One is that it is giving us, we have very precise ways now to gauge on how the vaccines work and how effective they are. And in fact, some of them appear to be more effective than actually getting the disease itself, which is pretty unusual, but it goes back to that. The vaccines are sort of designed to, these mRNA vaccines designed to initiate and have at least a surrogate of the disease happen according to the textbook immunological response to the disease. Okay, and like I said, many people don't have that, and that could be why the vaccines actually do better for long-term immunity. But I think we're learning a tremendous amount about infectious disease in general, and I think that is really changing the way that we are doing science, and it's really highlighted the issue of needing to look at lots of patients. I think in the past, when people would publish papers on 10 patients, and that's not really enough to. You can learn a few things, but you need to do large patient studies. I think team science has become important during this pandemic, and I don't think that's going away either.
Yeah, actually, I will also echo the similar thing. When I started using this multi-omics in infectious disease with HIV, it was very hard for me to do this analysis, because we don't know what kind of when bringing out of the biosafety level 3 what kind of material or other thing we required. So even I remember in a conference, I asked the core facilities of Sweden, okay, when we can think you can start taking infectious material. It's like not much before, it's like 4, 5 years before. And then now they realize this problem. I think understanding the infection biology is very, I think it's a long-term process. And when we have this technology that we can give a complete picture, not just the like one or two molecular, one or two pathways. So this is more and more valid. I can give an example like before COVID, we started this with the Crimea Congo hemorrhagic fever, which is biosafety level 4 pathogen. The cohort was actually in Turkey with one of my collaborator, and they transfer the sample. So initially, most of these biosafety level 4 pathogen are restricted to transcriptomics because you give trizol, you can take it out the infected cell outside of biosafety level 4. Now we have a protocol for proteomics because we know which buffer is safe to bring it out from biosafety level 4. And now we are actually validating protocol for metabolomics. So now it's where it's going on. So whether can we bring out the samples from level 4? So I think this COVID has give us wide opportunity to use more and more use of multi-omics technology in the infectious disease field. When that way, I'm happy because we struggle a lot in the beginning to get access to a proteomics facility or a metabolomic facility. So this is a long term, and for the COVID-19 as such thing, I will use the other way around. Still, 5% patient from both the trial, 5% patient are not responding, and it seems they are not only old people because even old people this 94%. So that means there are some younger people. So it will be very good to understand why they are not responding. So if you go in a negative way, why they are not responding to vaccine using this high throughput method, then it could be a much, we can learn a lot. And obviously this virus is new, so we have to understand a lot of virus biology. So that could be the one of the field too, because we are very much interested that what kind of metabolic reprogramming this virus do when it infect any cells. So that's what we are looking forward for.
We have a question from the audience that probably Jim can answer first, and then I think would also be a good one for Ujjwal, given the data. So what is known about the interaction between diabetes and the nutrition or food-derived metabolites in the COVID-19 patients that we're seeing? Kind of comorbidities question.
Yeah, so what we know is for me, right?
Both of you. So Ujjwal, if you'd like.
Okay, so I will take it as a positive way because all diabetic patients are not dying. So that means it's not a common factor. And there's a study, actually a report, it's not a study, came from the Swedish IVA. So there it shows it's not only one factor that is affecting, because even like people with more than 90 ages, they are 50, death is 50% or even low. So it's not a one factor. What we don't know, what is the factor? That could be a statistically associated comorbidities for the death, but there could be other reason, because we have a few cases of very healthy person without any underlying disease and this thing happen, there's a death. There's several doctors who are dead. So the more we understand this virus, or the more we understand this thing, it will come up. But one common feature between diabetes and this thing, what we have observed, that the plasma profile sometimes matches with the diabetic patient, like some cases. And so there are lots of new things to come up with the straight correlation between any comorbidity and death and severity.
Yeah, I think this issue of, I think comorbidities was a word that was not in the public vernacular until this year. And I learned what that means. And certainly the pandemic has greatly amplified the importance, but there's only a one or two comorbidities that actually seem to be really bad to have when you get COVID. There are others where your risk is higher. Diabetes is one. There are certain blood leukemias where basically they're extreme outliers in terms of the fatality rate. If you have what's called a CLL, which is a type of lymphocytic leukemia, it seems like your body simply doesn't even recognize you have the virus. And you go to a certain, and so those patients are at extremely high risk. In terms of things like diabetes or, obesity is an issue, cardiac conditions are an issue. I think that we are just now in a position to be getting to resolve those kinds of comorbidities and exactly what is the biology behind them. I think that's something that probably we're not going to have a good resolution on before the availability of vaccines. Vaccines are going to be coming out in December, January, throughout the next year. But it's going to deeply impact how we treat patients going into the future, because we're learning much, much more about those patients, their immune susceptibilities, et cetera. Things that we basically weren't studying before the pandemic, but have been highlighted by the pandemic. So, there is a silver lining to 2020. You have to look a little deep to find it.
Yeah.
But one of the silver linings is that we, of all the scientific community has really come together and collaborated around this pandemic. And out of that, we are actually developing a much deeper understanding of human disease in general. It's been a quantum leap, I would say. Before the pandemic, you could point to a couple of cancers that were probably the best understood of human diseases. Now, overwhelmingly, it's COVID-19, and lots of things we still don't understand. But I think comorbidities, patients that have those, we are really developing a much deeper understanding of how diabetes and other issues like that impact the human condition in a general way, mostly because of these broad approaches we've taken to understanding the virus.
I think we have one last question that, Jim, I believe you touched on before, but if you want to talk again about the idea of food sources and supplements being involved in being protective for COVID, or maybe making somebody more susceptible, or is it something that should be considered versus therapeutics or integrated into therapeutics for those who are ill?
Yeah, so I'm going to be talking I'm actually a chemist.
Excellent.
So you have to appreciate the fact that I'm not giving people medical advice here.
Yes.
Early in the pandemic, people began identifying, and if you look in the literature back, and it seems like decades ago, it was only like March and April and May, it's things like cytokine storms, out of control inflammation that was anticipating basically a death in many patients. And one of the treatments that was proposed was drugs that are used for similar cytokine storms in cancer immunotherapy patients that receive a certain type of cell therapy. And that drug seemed to work if you looked at the clinical metrics, like certain blood levels of analytes, et cetera, but it didn't make the patients better. So it didn't actually work globally. And my feeling is that it's because it addressed half the problem. You had these metabolites that were seriously depleted. At the same time, you had this pro-inflammatory environment, and you need to address both of these. That's my belief. And so I do think that there are dietary things that are relevant here, but I want to also add a note of caution. So we do see this profound depletion of lipids and amino acid catabolism. We're not the only ones who see this, of course, and Ujjwal, has some data. There's some groups elsewhere that have reported on some things like this. We also see a drop-off of xenobiotic metabolism. Xenobiotics are the metabolites you get from food. And when I say drop-off of metabolism, I mean that the upstream things like the direct food products you eat actually go up a little bit. It's all the downstream products that go down. And meaning that you're just stopping that metabolism. That could point to liver damage because that's where that happens. But it also could point to the fact that you could provide supplements to the patients, but they may not be processed. There's a lot of dysregulation that we don't understand yet, but we're getting a window into. So I do believe ultimately that we will figure out how to balance drugs and non-therapeutic, non-pharmacologic interventions. I think supplements and things like that are actually very important, but there's a lot of unknown questions as well. So that's the message I wanted to get across.
Well, thank you. I don't know if you have any final thoughts, Ujjwal, as we're wrapping up.
Yeah, so I think the base part of the COVID thing, if we go take it in a positive way, that it has like literally forced us to work as a team. So several disciplines come together to find out the clue, which is enormous, because this was sometime earlier shown in cancer field or lifestyle-related disease, but not that much in infectious disease. So that is a one strength that the scientific community has shown that. So they are coming up, immunologists, molecular biologists, system biologists, chemists, and virologists, including the clinician, so to have a meaningful outcome to understand this virus. And I think, and on a positive note, again, like we now know how to react, because I was looking for Ebola when Ebola break out this thing. There's one paper which has used actually this kind of multi-omics technology to find out how Ebola infect or how spread. So this is the positive side of this thing, and hopefully we'll have the vaccine soon, and then this go up. But it will teach us how to behave when this kind of pandemic happen, and how to react to those kind of unknown, or because if we see that it's like only 6 to 8 month time that we know lots of things about this virus. It could not have been possible if the scientific community do not come together. So that is the my take-home message, that it has teach us also something that we can go together to achieve the best.
Jim, last thought?
I think I said most of what I want to say, but
Yeah.
I hope 2021 is a better year.
Yes, yeah. I think we can all agree. Well, I want to thank you both so much. So Jim, it was great to have you back for a follow-up conversation, because we had spoken, I think, back in May when you had started on this. It's so exciting to see.
Like years ago, doesn't it?
Oh my goodness, does it ever? So it's great to see the work that everybody's doing. It's really gratifying to see, being an employee of Metabolon, to see how useful the data that we provide can be integrated to answer questions, and then also, provide more areas of research. And I think my take-home message is I'm very heartened by the idea that a lot of what is being done now with all of these different data streams may speed up the response to the next pandemic, because we know there will be a next pandemic. So those are my final words. But again, thank you both so much. This was really wonderful.
Thank you.
For sure. Bye-bye.
Bye.